Good morning, everyone. I'm Hongjin Zhou from Shandong Provincial Hospital of China. It's my great honor to share some viewpoints about COVID-19. My topic for today is pharmacological rationale for antihypertensive drug choice on COVID-19 affected patients. ACER and ARB might not increase their susceptibility. COVID-19 is caused by severe acute respiratory coronavirus 2, in short, SARS-CoV-2. This virus spread mainly through the respiratory route. That's why mask is very important for the prevention of the disease. And the virus use ACE2 as a receptor for infection. On March the 11th, 2020, WHO declared the global pandemic of this disease. Therefore, a lot of efforts have been made to the research about this disease, especially the receptor ACE2. ACE2 was firstly discovered in 2000. They are widely expressed in lungs, cardiovascular system, and kidneys. ACE2 has a transmembrane domain as well as an extracellular domain. And this is a claw-like proteinase domain by which it can bind to the receptor binding domain of spike protein from SARS-CoV-2. And with the help of this binding, ACE2 can mediate SARS-CoV-2 into the cells for replication. SARS-CoV-2 and SARS-CoV in 2003 share the same receptor, ACE2. However, the binding affinity between SARS-CoV-2 and ACE2 is much higher than that of SARS-CoV, which might account for the greater pathogenicity of SARS-CoV-2. Besides as a receptor of the coronavirus, ACE2 functions in RAS system. This diagram shows the RAS system. Angiotensinogen is converted into the angiotensin 1 with the help of renin. And angiotensin 1 can be converted into angiotensin 2 with the help of ACE, angiotensin converting enzyme. And then angiotensin 2 can bind to the angiotensin type 1 receptor for inflammatory function and vasoconstriction. And the engine 2 can be catalyzed by ACE2 to angiotensin 1,7, which can then bind to the mast receptor for anti-inflammation. So there is a balance between ACE, angiotensin 2, and the angiotensin type 1 receptor. This is for inflammation, and another is an ACE2, angiotensin 1,7, and the mast receptor. This is for anti-inflammation. So there is a balance between them. And the ACEI can inhibit the activity of ACE. In this way, it can reduce the production of the angiotensin 2, while ARB can inhibit the function of angiotensin 2 through blocking the binding of the angiotensin 2 and the angiotensin type 1 receptor. angiotensin 2 function play a great role in hypertension and myocardial remodeling. So ACR and ARB have been extensively used for hypertension and heart failure through inhibiting the production or function of angiotensin 2. However, some papers show that ACEI and ARB also can upregulate ACE2 expression. ACE2 is also used as a receptor COVID-19. So there is a question: ACR and ARB, whether ACR and ARB can increase the susceptibility or severity of COVID-19 through upregulating ACE2, or whether ACR and ARB should be converted into the CCB for hypertensive patients during COVID-19 pandemic. In other words, higher ACE2, whether higher ACE2 stands for higher susceptibility and severity of COVID-19. With this question, we did a lot of literature review. And one paper shows that as the time the lower expression and activity of ACE2 in aging women. A mouse experiment also shows 3 months in mice is similar to like a younger adult, and 12 months is similar to middle-aged person, and 24 months is similar to old person. Yeah, something like that. And this experiment shows that in female mice, the expression of ACE2 reduced as aging. The same situation occurs in the male group. So this figure shows that ACE2 expression reduced as aging. So whether severity of the COVID-19 is also reduced as aging, there is another paper, published in New England Journal of Medicine, and we get some data and did another analysis. For example, this paper, this clinical research recruit 1,011 patients. 9 patients from this group and 153 patients come from this group. Then we get this data from and then write it down here, the total patients in different groups, in different groups, and then the severe cases from here is the 1 severe cases from this group, and write down it here, and then another and the other group, the other cases, and write down here, and then we got the severe ratio, and we found that the 11, 12, and 17, and especially in this group, the older patients have a higher severe cases. The ACE2 expression reduced as aging, but severe COVID-19 cases rise along with aging. So that means which is reversely correlated to the ACE2 expression, right? So that means higher ACE2 expression doesn't mean higher severity, more severity of COVID-19. How about the susceptibility? Whether higher ACE2 is equal to higher susceptibility of COVID-19. The same as it's also come from the mice experiments. 20 months means old person group, and this figure shows that this figure suggests that old women have higher ACE2 levels. Expression level higher is significant, so it's from the mice experiments. But however, SARS-CoV-2, SARS-CoV-2, share the same ACE2 with SARS-CoV 2003 shares the same receptor ACE2 as SARS-CoV-2, right? It's from the previous clinical research, we found that SARS-CoV-2 preferred old men. So that means old women have higher levels, but SARS-CoV-2 prefers old men. That means high ACE2 doesn't equal to high susceptibility of COVID-19. So what's the reason for that? Because the ACE2 has two forms, membrane-bound forms and the soluble forms. And the membrane and the only membrane-bound ACE2 can bind to the SARS-CoV-2 and then mediate the SARS-CoV-2 entry into the cells for replication. The membrane-bound ACE2, can be cleaved by the ADAM17 to become the soluble one. The soluble form, soluble ACE2. The soluble ACE2 can bind to the SARS-CoV-2, but it can mediate the coronavirus invasion into the cells. On the contrary, it can bind, the soluble ACE2 can compete the membrane-bound ACE2, compete the SARS-CoV-2 with the membrane-bound ACE2. It can bond to the coronavirus and stop the binding of the coronavirus with the membrane-bound ACE2. So in this way, soluble ACE2 plays a protective effect. And several papers also confirm confirm the protective effect of soluble ACE2. One paper, just recently published in the Cell Journal. In the cell cultures, infected with the SARS-CoV-2, they found that the human recombinant soluble ACE2 can inhibit SARS-CoV-2 infection in dose-dependent manner. However, the mouse recombinant soluble ACE2 can't function in a similar way, such implicating the species specificity. They also established the human capillary organoid from human pluripotent stem cell, and then established the human capillary organoid. Anti-CD7 means marks the endothelial cells, and PDGFR beta, marks the parasite cells. And with these organoids, they also proved that the human recombinant soluble ACE2 also inhibits SARS-CoV-2 infection. As is known, during the pathological process of the COVID-19, the binding of the ACE2 and the coronavirus consumes a lot of ACE2 molecule, and leading to the downregulation of ACE2 molecules and upregulation of the angiotensin-2, exacerbating the pneumonia and hypertension. That means the angiotensin-2 angiotensin type 1 receptor is increased, while the ACE2 angiotensin 1 to 7 and the MES receptor pathway is reduced. And in one severe patient, researchers, treat the patients with a clinical grade human recombinant soluble ACE2, and the red triangle stand for the days on which the drug was applied. And they found during the drug administration, the angiotensin 2 is reduced, right? And also the angiotensin 1,7 is upregulated, right? That means the balance, the imbalance return to balance. ACE2 is upregulated. The soluble ACE2 can upregulate this pathway while downregulate this pathway. And they also found that during the human recombinant soluble ACE2 administration, the inflammatory biomarkers such as IL-6 and TNF-alpha are also reduced as days going. That means the soluble ACE2 really has a clinical protective effect on COVID-19 patients. If ACR and ARB also, if ACR and ARB also increase the soluble ACE2, it also can plays additional protective effect through this way, soluble ACE2 upregulation. Besides there's the effect on the ANG2 reduction. It also can exert additional protective effect through upregulating soluble ACE2. And also several clinical research also confirmed that ACR and ARB didn't increase the susceptibility of COVID-19. It's published in New England Journal of Medicine. And also ACR ARB can reduce the all-cause mortality in hypertensive COVID-19 compared with the ACR ARB non-users. They confirm the protective effect of the ACR and ARB on the COVID-19. So, the question for the hypertensive COVID-19 patients, which is better, ACR or ARB? So return to this diagram. As it seems, ACR can inhibit the ACE-mediated ANG2 production, but it has no effect on the non-ACE2 pathways. The non-ACE-mediated angiotensin 2 production, such as chimers, cathepsin, and also, however, the ARB, can functions through blocking the binding of the angiotensin 2, to the angiotensin type 1 receptor. And no matter how this angiotensin 2 is produced, through ACE-mediated pathway or non-ACE2 pathway, it can block the function of, it just blocks the function of the angiotensin 2, and then transfer block this bonding and help convert this angiotensin to this pathway, or to bind the lower affinity type angiotensin type 2 receptor. In this way also functions for anti-fibrosis and pulmonary hypertension, and also it can transfer a lot of angiotensin to go through this way, functions anti-inflammation. Moreover, ACEI also can inhibit the ACE2 mediate bradykinin degradation. That means a lot of bradykinin will be accumulated when using ACEI, and the bradykinin will bind to the BKB2R for the pro-inflammatory action, will exacerbate the inflammatory situation. However, several ARBs such as valsartan, it's reported that valsartan can bind to the PPAR gamma, functions anti-inflammatory actions. So in this point, ARB may superior to ACEI in theory. Of course, this theory also need to the clinical research to confirm that. Thank you for your attention. This is all what I want to share.